Hello everyone. So today I'm going to talk about malaria. Here we have three scientists, Alphonse Lavrin, Sir Ronald Ross, and Miss Tu Yu. So the three of them got the Nobel Prize. Now Nobel Prize for what? Alphonse Lavrin, he discovered plasmodium in RBCs of a patient in Algeria. Sir Ronald Ross, he described the sexual cycle of the parasite in female Anopheles mosquito in Sikandarabad, which comes in India. Miss Tu Yu, she is a chemist and she got the Nobel Prize because she discovered artemisinin. Now this artemisinin is a drug which is used for the treatment of falciparum malaria. Okay, now let's talk about the agents for the human infection. There are 5 agents in total. 4 of them we already know, and the fifth one is recently discovered. Now, all the 5 are Plasmodium vivax, Plasmodium falciparum, Plasmodium ovale, Plasmodium malaria, and the recently discovered is Plasmodium knowlesi. Plasmodium vivax causes benign tertian malaria. Falciparum causes malignant tertian malaria. Plasmodium ovale causes ovale tertian malaria. Plasmodium malaria causes benign cotton malaria, QUARTAN, cotton malaria. And Plasmodium knowlesi, it causes quotidian or simian malaria. Now this Plasmodium knowlesi, it is a parasite of monkey, but it can also infect humans. Okay, so the Plasmodium vivax, in this the fever occurs once in 48 hours. In all these three, the first 3, fever occurs once in 48 hours, that is, it recurs every 3rd day. In Plasmodium malaria, the fever occurs once in 72 hours. And in Plasmodium knowlesi, fever occurs once in 24 hours, that is, it recurs every day. Okay, just keep this in mind, this is of prime importance. Now let's talk about the life cycle. Now Plasmodium completes its life cycle in 2 hosts. It requires a definitive host and an intermediate host. Now, as we already know, the definitive host is female Anopheles mosquito and the intermediate host is the man. In the female Anopheles mosquito, it occurs through the sexual cycle or sporogony. In man, we have asexual cycle or schizogony. Now the male Anopheles mosquito, it feeds on fruit juices, so that is why it is not responsible for the transmission of the disease. While the female Anopheles mosquito, it needs at least 2 blood meals before laying eggs, so that is why this one is responsible for the transmission of the disease. Got the difference between the two? Okay. Now let's talk about the vectors of primary importance. We know that the Anopheles is the vector. Anopheles is the species which causes the transmission, but we don't know that we have more vectors, that is Anopheles, Culicephasis, Anopheles stephensi, and Anopheles fluviatilis. These are the three vectors. Culicephasis, it is found in the rural areas. Stephensi is found in urban areas, and fluviatilis is found in hilly areas, in hilly regions. Okay. Now let's talk about the human cycle, the mode of transmission and the infective form in it. Now the human cycle occurs asexually. Okay, so what happens is, when the female Anopheles mosquito, when it bites us, in its salivary gland it contains sporozoites, which are the infective forms. So when it bites us, these sporozoites are transmitted into the cutaneous venules, and then it enters the blood circulation. Now in this, the infective form is sporozoite. Okay, now rarely what happens is, it is transmitted through blood transfusion or transplacental transmission. So in this, the infective forms are the trophozoites or merozoites. So this is the difference between the two, but this occurs very rarely. Most common one is the sporozoites, which occurs through the bite of the female Anopheles mosquito. Now in human cycle, in humans, the asexual cycle occurs in 3 stages, that is pre-erythrocytic schizogony, and gametogony. Now let's talk about the first one, that is pre-erythrocytic schizogony. It occurs in the liver, that is also called as hepatic. So it occurs before the invasion of the RBCs. It is also known as exoerythrocytic stage, or intrahepatic stage, or tissue stage. Okay, these are the 3 names for this stage. Now what happens is, the motile sporozoites, they leave the circulation within 30 minutes and it enters liver. Now this sporozoites, spindle shaped sporozoites, what happens is, they contain a protein which is called circumsporozoid protein. This sporozoites, on the surface of it contains a circumsporozoid protein. So when it binds to the receptors which are present on the surface of the hepatocytes, after its binding, these sporozoites enter the liver. Then after entering, what happens is, these sporozoites, they are transformed into trophozoites, which are rounded. Okay, the spindle shaped sporozoites are transformed into rounded trophozoites. Now these trophozoites, these are the feeding stage of the parasite. This is the feeding stage of the parasite. So it undergoes several nuclear divisions, and it transforms into pre-erythrocytic schizont. Then this pre-erythrocytic schizont, it contains several merozoites. it contains several merozoites, and when the hepatocytes rupture, these merozoites are released outside. Okay, and then these merozoites, they attack the RBCs to initiate erythrocytic stage. Now the key point in this is that in malaria there is no liver injury because plasmodium affects very few hepatocytes, so hepatic damage does not occur in malaria. This can come as an MCQ. So talking about the duration, it can vary from 5 to 15 days depending on the species. Now what are hypnozoites? As we have seen that the sporozoites, which are spindle shaped, they transform into trophozoite, right? But in some species, that is Plasmodium vivax and Plasmodium ovale, what happens is they do not develop further and they remain in the liver as hypnozoites. So these two remain in the liver as hypnozoites. Fine, and they cause relapse of malaria after many years. Then what is the difference between relapse and recrudescence? They are differentiated from each other. See, relapse and recrudescence, they can be differentiated from each other by this, that the relapse occurs in Plasmodium vivax and ovale, and recrudescence occurs in Plasmodium falciparum and Plasmodium malaria. Okay, recrudescence occurs in the malignant forms and relapse occurs in vivax and ovale. Okay, now we are done with the first stage, that is pre-erythrocytic stage. Now let's talk about the erythrocytic schizophrenia. Now the hepatic merozoites, after the rupture of the hepatocytes, when these merozoites are released on its surface, what happens is these merozoites, they attack the RBCs. Now on the surface of the RBCs we have glycophorin receptors. These merozoites bind to the glycophorin receptors, they enter by endocytosis, and within the inside the RBCs we have a parasitophorous vacuole. So these merozoites are contained within it. Now after that, what happens is this merozoites, after its entry into the RBCs, they are transformed into trophozoites. Okay, then we have two forms, that is early trophozoite and late trophozoite. These early trophozoites are also called ring forms because they are annular or signet ring in appearance, and late trophozoites, the ring form enlarges and becomes more irregular, and therefore it is called as amoeboid form. Okay, so in early trophozoite is called ring form because its shape is annular or signet ring in appearance. It contains a central vacuole with a peripheral thin rim of cytoplasm and a nucleus. Okay, and in this late trophozoite, the ring enlarges, and it enlarges and it becomes very irregular, and it transforms into amoeboid form, which is also called late trophozoite. Okay, then after that we have the malarial pigments. Now this plasmodium feeds on what? It feeds on hemoglobin. So after it feeds on hemoglobin, the undigested products, that is hematin and iron porphyrin. Hematin and iron porphyrin, these are the malarial pigments. Malarial pigments, or it is also called hemozoin pigment or hemozoin pigment. So these are the undigested products. Now what happens is we have seen the late trophozoite, which is irregular or amoeboid shape. Okay, so this late trophozoite undergoes schizogony and it is developed into 6 to 30 daughter merozoites which appear in the form of a rosette. 6 to 30 daughter merozoites which appear in the form of a rosette, and this form is known as erythrocytic schizont. Now this RBC, it contains daughter merozoites, malarial pigments and toxins. What this RBC contains now? Daughter merozoites, toxins and malarial pigments. So these substances are released into the circulation and they cause malarial paroxysm of fever. After it's released into the circulation, they cause malarial paroxysm of fever at the end of each erythrocytic cycle. Then these merozoites enter into the RBCs and then the cycle is repeated. Now here the point to remember is intraerythrocytic life cycle varies from 48 to 72 hours depending upon the species. In Plasmodium falciparum infection, the later stages of the erythrocytic cycle, that is trophozoites and schizonts, they occur in the capillaries of the brain and internal organs. Okay, so when we do the peripheral blood examination, only the ring forms are found in it. Only the ring forms can be found in the peripheral blood examination, and the late trophozoites and schizonts are not found because the later stages occur in the capillaries of the brain and internal organs. Okay, this occurs only in Plasmodium falciparum infection. Now here we are done with the second stage also. Now let's talk about the third stage, that is gametogony. After a series of erythrocytic cycles, this merozoite, it enters the RBC. After its entry into the RBCs, it is not further developed into a trophozoite, but it transforms into sexual forms, and these sexual forms are called as gametocytes. These gametocytes are rounded except in Plasmodium falciparum. In it they are crescent or banana shaped. This thing we already know. Now these gametocytes are of two types: male gametocytes and female gametocytes. Male gametocytes are also called as microgametocytes, and female gametocytes are also called as macrogametocytes. Now these gametocytes, they do not cause Neither they cause any clinical illness. They do not cause any clinical illness, nor they divide. But its important role is transmission of disease. Transmission of disease. These gametocytes are responsible for the transmission of the disease. Gametocytes, these are the infective form to mosquito. Infective form to mosquito. They are capable of transmission only when they are mature, viable, and present in sufficient density. Okay, that is 12 gametocytes per cubic mm of blood. So this was the human cycle. Now let's talk about the mosquito cycle. It is sexual cycle. Now this female Anopheles mosquito, during blood meal, it takes up asexual as well as sexual forms. So these asexual forms, they get digested, and the sexual forms, that is gametocytes, they undergo further development. Okay. Now the male gametocyte and the female gametocyte. Male gametocyte undergoes X flagellation and it forms 8 flagellated actively motile bodies, which are called as male gametes or microgametes. After X flagellation, what it forms? 8 flagellated actively motile bodies. And the female gametocytes, it does not undergo X flagellation. It does not undergo X flagellation. It directly undergoes it directly forms one female gamete or macrogamete. Okay. Now, as we already know how the zygote is formed, it is formed by the fusion of the male and the female gametes. So when this zygote, it transforms into motile and elongated forms, it is called as ookinete, and which occurs in the midgut. Okay. Now these ookinetes are motile and elongated. When this ookinete penetrates the stomach wall of the mosquito, this is the mosquito. When it penetrates the stomach wall of the mosquito, it is transformed into a rounded form, which is called as oocyst. Now this oocyst contains a thin elastic membrane on the outer side. Okay. So when this oocyst undergoes sporogony, that is meiosis, it forms 4 spindle-shaped sporozoites. These are the sporozoites. When this mature oocyst ruptures, these spindle-shaped sporozoites, they are released and they migrate towards the salivary glands of the mosquito. Okay, these sporozoites migrate towards the salivary glands of the mosquito, and the cycle is repeated. Fine, so this was the mosquito cycle. Now the extrinsic incubation period, it varies depending upon the species, so it can vary from 1 to 4 weeks. Okay, नहीं ठीक है. So the extrinsic incubation period, it depends, the varies from 1 to 4 weeks depending upon the species. Okay, so that's all about the life cycle. Just let me know in the comment section if you need any changes in the video, or if you need extra information or whatever video you want. Just let me know what all things I can add up to it. Okay, thank you so much.